part we will learn about the polysaccharides. Okay, polysaccharide means more thousands of mono units are present there, right? So on the basis of mono units, we classify polysaccharide into two parts, homopolysaccharide and heteropolysaccharide. In homopolysaccharide, we will see the same type of mono units are present. Like cellulose, it is made up of same type of glucose. It is made up of glucose. So many glucose molecules, thousands of glucose molecules made cellulose. Okay, so one type of mono units are present here. Heteropolysaccharides means if the mono units are different, like in hemicellulose, fructose, xylose, mannose, albinose, this type of different mono units from this polysaccharide. So this is called heteropolysaccharide. Okay, we can classify polysaccharide also on the basis of their function. So on the basis of their function, there are two types of polysaccharide. One is storage polysaccharide, one is structural polysaccharide. In the storage polysaccharide, the example will be starch, because we know now in plant body starch is stored, right, as a food. Glucose is stored as a starch, the storage food. And the structural polysaccharide cellulose, the cell wall in plant cell, cellulose is cell wall is made up of cellulose, it is structural polysaccharide. So one by one we will learn their structure also. Starch we will learn in details, then cellulose also we will learn in details. First we will learn starch. Starch is a monopolysaccharide and also is a storage polysaccharide, right? So it is made up of glucose unit. There are two structures are present in starch, that is amylose and amylopectin. In amylose, 200 to 2000 number of glucose molecules are present. In amylopectin, 2000 to 20,000 glucose molecules are present. And these are the two structure of starch. Okay. In amylose, we will see which type of bond present between this glucose molecule, 1,4-glycosidic bond that we have already learned here. 1,4-glycosidic bond. So here will be another, here will be another, here will be another. So the structure will be linear, right now, because one glucose, again between 1 and 4. So this is a linear. So it doesn't mean it will be a straight chain. It may be in this way, it may be in this way, it may be in this way, whatever the thing, but they must be linear. But they are present in very coiled manner. So they are highly coiled, but they are linear structure. Okay. But in amylopectin, we will see 1,4-glycosidic bond is present, but 1,6-glycosidic bond is also present. It means it is with 1 carbon, 1 and 6 number carbon, the bond is forming. With 1 and 4 number carbon, the bond is forming. So between these two, the bond is forming. And between 1 and 6, you see, this is 1, 2, 3, 4, 5, 6, the bond is forming. Clear? So this is a branch structure we can see, right? So the structure is not linear. This is a branched like this. Okay. Now in starch, what is the ratio of amylose and amylopectin is present in starch? It is present in 1:3 ratio. And why we know now starch gives blue-black color in iodine solution? Why? Because of this amylose. So blue-black coloration due to the amylose. Now starch, we know starch is stored in different parts of plant, right? In tubers of potato, we see starch is present. In maize also starch is present. So how they present? So they are present in two different forms. One is simple starch grain, we can say. One is compound starch grain. In simple starch grain, this is the hilum, and within this is the hilum is on one side, and the starch molecules are present like this. So this type of arrangement we see in maize. And if there are many hilum and the starch layers are around this, so this type, this is called the compound grain. We see this in rice and potato. In simple grain, on the basis of the hilum, In simple grain we have learned, we can again classify the simple starch grain into two categories. If the hilum is at one side, that we have learned now, the examples are maize, I told you. And if the hilum is at center and the starch layers are around it, then it's called concentric and it's eccentric. Okay, so how starch is present, their arrangement we have learned. Now the most important thing you remember, plant synthesis carbohydrate as glucose. We know plant synthesis carbohydrate is as glucose. In photosynthesis we have learned this glucose is a reducing sugar. Glucose is a reducing sugar because it has a aldehyde group, and highly reactive, right? Because reducing sugar and highly reactive functional group is present. So when it is transported, it is converted into sucrose. So when it's transported, glucose, another thing, so it will be as sucrose, it will transport. And sucrose is a disaccharide but non-reducing, because sucrose is from glucose plus fructose. It is a non-reducing sugar because both the functional group form the bond. There is no functional group, that's why it is a non-reducing sugar, and it is not going to react with anything. So N is not going to react with anything, and starch and stored as starch. So it will be stored as starch. Clear? So in this way, glucose is converted, transported as sucrose but stored as a starch. Now you can ask me, ma'am, why it is not, when starch will break, it will form glucose. So why it is not storing as glucose? Because glucose is a highly reactive, because it has a reducing power. Right, it is highly reactive. It is not a stable compound. But starch is non-reactive and it is stable. And another thing we know, in glucose, the bond is forming by removal of hydrogen molecule in glucose. Right, the bond is forming the removal of one hydrogen molecule. And if I tell 1000 glucose molecules are there, so how many water molecule will remove? 999. Water molecule have to be removed to form 1000 glucose molecule in a linear manner. So there is a lot of loss of hydrogen and oxygen. That's why it is stored as a starch. Okay. And how the starch break and form glucose? By adding hydrogen. So starch adding hydrogen, it will form glucose. Hydrogen means the water will break the bond and it will form glucose. Okay.